you know, some of these standards really, there's a level of compliance that you have to stay at, but you can develop prescriptive methodologies that fit your intended purpose. It really doesn't help you, I think, from developing standards for your own purposes. So what it does mean then is that you will need to develop standards and detailed methods for tissue collection and use that are appropriate for your intended end use if they do not exist. This is a figure I created with my marketing guy about 6 months ago, and part of the reason why I created this was to try to communicate to a lot of the stakeholders that I interact with, researchers, cell therapy companies, medical device companies. There are all these upstream things that on the sourcing side you may not pay attention to, you may not pay attention to the standards, but you either should or you should find organizations that do this, because a lot of these different factors, and I'll take you through the specifics of each of this on following slides, can affect the downstream performance and quality of your products. Whether your tissue comes from an old donor, young donor, male or female, is race important? Is gender important? Your sampling from heart, does it matter whether it's left ventricle or right ventricle? A lot of clients that come to us that are sourcing tissue for their particular need, they're not really thinking about all these things, and then they get something that doesn't perform in a dish the way they would like it to. So my story here in part today is a research community. We need to think a little bit more about human variation and its impact, and not even just human variation, but the processing side of things, what these source sites are all doing. If you source from pathology, for example, and your specimen's sitting there for 3 hours before they provide it to you, you know, what do you think is going to happen with that tissue or the cells you're trying to isolate? So again, things to be mindful of. I'm going to switch gears and talk a little bit about a project that we were involved with for about 5 years called the Genotype Tissue Expression Project. Some of you may be familiar with the program. This was the brainchild of Director of the NIH, Francis Collins. It was a NIH Common Fund eventually funded project, a lot of stakeholders involved in it, but I really see this as a case study and a great story of a group of individuals that came together with a common vision, much like you guys, that said, you know, we have an idea of something that we want to do, and there's no standards out there to tell us how to do this. So the various stakeholders that were involved in this study sat down prospectively, again, much like what this working group's doing, they designed with the end in mind. And it was a wonderful study, it's a wonderful story, and it was actually very successful. So their goal was to develop and standardize methods for collecting and analyzing 34 or more human tissues from over 900 human donors. And that could be used for gene expression studies, ensuring that data was valid and reproducible. So a pretty daunting task. Also to establish a resource database and associated tissue bank for the scientific community to study the relationship between genetic variation and gene expression in these human reference tissues. So again, a lot of stakeholders involved. I probably missed a few here, but from top down, Office of the Director, Francis Collins, NHGRI, BBRB from NCI was involved, Van Andel Research Institute, the Broad Institute, University of Miami Brain Bank. They did all the isolation of brains that were sourced for the study. NDRI, of course, was involved. And we also worked with about 6 organ procurement organizations. And we had to standardize this, and we had to do it across all these sites. So I'm going to stop there, and I'm going to show you a clip. I hope it works. NDRI recently had an awards ceremony. We presented a Service of Science Award to Francis Collins for his obvious contributions to science. But it's a good clip because it's, rather than hear it from me, you'll hear it from Francis why the GTEx project was so important. I don't know, do I need to start this or can you start this? No sound. Sorry, I thought they worked on this earlier. This is a short clip anyway, it's only 2 minutes. No luck? Well, Well, I'm sorry about that. That's okay. Again, the slide, if we can't get it working, the slide decks will be made available to everybody. It's just a compelling story of Francis telling the story of, you know, how they came up with this idea for GTEx and why it was a daunting task, and in the end they were successful in standardizing what they needed to get very good data at the end of the day. Just a schematic of the experimental design. This is really how the pilot study was set up. There were surgical donors, and it says autopsy donors here, but they were really organ donors and tissue donors. 50 or more tissues from each donor was collected initially. Eventually, in the scale-up phase of the project, they reduced that number to about 35 tissues. Tissues were harvested from these donors, either for histopathology. Some were plunked in PAX-Gene and for RNA purification, and ultimately sequencing. Again, a very, very large and daunting study. You can imagine standardizing this, developing SOPs, communicating across all these stakeholders, and then training them to make sure they could do it was quite a task. But they did it. Just give us sort of a background of the donors that were involved in the study or contributed to the study. Most of the donors were 48 years of age or older. Most were Caucasians. I won't get into the specifics of that, but there's cultural and religious reasons why I think other ethnicities and populations don't typically consent for donation. Again, but one of the problems and complexities that we have to deal with in human tissue sourcing. RNA integrity, of course, because the end use was to do DNA sequencing, or I'm sorry, RNA sequencing. The integrity of the DNA was very important for the project. So quite simply, two different plots here: RNA integrity number on the y-axis, postmortem interval of time on the x-axis. Blue and green dots are organ donors, and the red dots are tissue donors. And the left side is PMI and RIN numbers from the heart, and the right side is for colon. Take-home message was, and probably no surprise to everybody in this room, there is a correlation between the RNA integrity and the amount of time that organ or tissue has been out of the body. Interestingly, though, we did not see that for brain. Project outcome: there were 900 human donors, 13 surgicals that had consented and tissues recovered from. About 80,000 biospecimens collected. About 96% of these samples were released to BBRB for their deposit in their cancer human biobank. RIN numbers were actually very good. About 84% were over 7, so higher numbers better. Consent rate was actually pretty high for this study. You'd be surprised how motivated families were to participate. PMI average, so this is for organ donors, was actually pretty low. It was pretty good, 3.5 hours. So all those SOPs and standards that were developed for the study are actually sitting at BBRB, and this is a link. So if anybody ever wants to go on and look at how they did it, that information's there. What you can do. So like GTEx, have detailed standards and methods for your specific end use, and uses do not exist. Work within your discipline to develop it. It's not an insurmountable task, and the GTEx story, I think, is a pretty convincing one that it can be done. Make sure your supplier's consent forms capture the intended use of the specimens. Many of you may be aware of a lot of to-do around common rule in the last 2 or 3 years and what consent and authorization should cover. It's moving in that direction where more is better. So if you're going to do genomic studies, work with a sourcing partner that can ensure that you've got consent language that covers that. If you can audit or visit your suppliers, you will learn quite a bit, whether you walk down to pathology and you see how they're processing, or if something comes down from surgery and you see how long it sits, where it sits, how it's been used. All this stuff's going to inform you into maybe how you should do things differently or a little bit better, or how it's potentially affecting your data. Use of your specimens should dictate your donor source type, PMI requirements, etc. Again, designed with the end in mind. Consult your sourcing partner when planning your study. Some of the questions that we will ask is, again, I come back to donor eligibility. Are age, race, and sex important to your study design? Think about that. presence or absence of disease, comorbidities. These, again, are things that we have to screen for if you need it. Genotype, so we're working with the CF Foundation and Vertex Pharmaceutical right now, and this information is in public domain, so I can talk about it. We source CF lungs with specific haplotypes that they're interested in for drug screening purposes. And so now we have to make sure that our consent language covers that, and the patients are aware that there's always that risk of potentially being identified because you've disclosed that genetic information. Build your study protocols like you would a clinical trial protocol. Inclusion criteria, exclusion criteria. Think about what you need, why you need it. Design the perfect study as much as you can. Control what you can control. Procurement. Time from death or tissue collection to preservation and use. Like GTEx, RNA integrity numbers for sequencing was really important. They really needed access to organ donors for that study. If you're doing something that's more histopathology-minded, maybe not so much. Living or post-mortem. Again, post-mortem tissue might be fine if you're sourcing from an organ donor. Again, for some studies, tissue donors are fine. We get a lot of requests from investigators that want information from or samples from living individuals. Let's get more on the biomarker side of things. Does your organ or tissues need to be perfused or flushed? Another question that we get asked from time to time. Or we ask in turn the researchers if it's important to them. Tissue sampling, homogeneity, important. Again, we have investigators that come to us, are isolating hepatocytes for some sort of work that they're doing. Well, does the lobe of the liver important to whether that's, the hepatocytes are in proximity to the vessel, is that important to you? Again, using my heart example, do you need left ventricles different than right ventricles? So think about all these things and delineate as best you can what you need for your studies. It will affect your results. Method of preservation required. Again, we have companies that come to us all the time. I want to source cells. I'm going to ultimately develop them for X, Y, and Z. They have no clue upstream what they need to. So the organ side of things, they've got transport media like HTK, UW, things like that work and preserve organs pretty well. But you as researchers that are doing cell isolation work, think about how that tissue is going to be affected as it sits and what works out best, what would work best for your end use. So do you want it sitting in a cell culture media with antibiotics? Do you want it transported just cold, not flushed with anything? Again, these decisions are all going to impact your experimental results to some extent. Level of severity required, again, depends on your end use, but there are things that we need to know. Level of compliance required by your suppliers. So again, if I have a cell therapy company that comes to me, it's going to develop a product as a therapeutic, I have to make sure our quality assurance group is auditing sites with GTPs in mind. It's a different level of compliance. They're going to have to be able to, or we are as the sourcing provider, convince the FDA and their CMC that we've covered all the aspects of that level of compliance. Challenges to be mindful when sourcing human tissue. Some other things to think about. Healthcare organizations' primary objective isn't to provide organs or tissues for research and development. No surprise. Infrastructure for tissue acquisition management in healthcare settings is not well defined. Some hospitals do a good job of it. They're very research friendly. They might even have a procurement group. Others are not. Finding the right stakeholders to work with is often difficult to navigate. You're going to go to the surgeon. You're going to go to the head of surgical pathology. You're going to go to the chairman. You need to find out who the right person is in the hospital to access what you need. Legal and regulatory tissue acquisition and use for research is currently not regulated in the U.S., and the laws around human tissue procurement and distribution, they vary from state to state. New York, for example, you cannot procure and distribute tissues unless you're a licensed tissue bank within the state of New York. Now, again, we're not physically located there, but because we're licensed to the state of New York, we can do this. But that's not so the case in a lot of other states. Vast phenotypic heterogeneity across human donors, no surprise, but again, something to kind of work into your studies, or at least be mindful of. Cost of keeping a recovery team on site to procure for research is often not practical. Something we deal with a lot on the OPO and tissue bank side. They're sending in recovery teams, they're going into an OR, they're collecting tissues. Again, you can't get in the way of a transplant, but for them to keep a team there longer than the transplant, the organs coming out need to be, excised and recovered and packaged. Their costs that they incur by keeping those people around, we in turn, they're not going to recover one piece of tissue for us if they have to keep a whole team there. So something that is just sort of a constant challenge that we have. So working with organizations that are doing multiple procurements might be another way, easier way to get what you need. So there's going to be times that approaching your OPO works great, and there's times when it doesn't. Most postmortem organs or tissue recoveries happen in hospital settings. Again, I just covered that. Most OPOs aren't set up to accommodate research requests, but they're getting there. Some are more research friendly than others. I know some of you in the room work directly with your local OPOs. That's great. If I'm in Palo Alto, California, I'm not going to contact Gift of Life in Philadelphia. They're probably not going to help me. Your local OPOs will help you. They do get some sort of credit from CMS for research allocations as well. So explore those as options for tissue sourcing. Researchers are not always willing to accept organs or tissues 24/7. So I have a researcher comes to me, they're interested in getting a heart for a study, or maybe probably a bad example, Parkinson's brain. We explain to them, the OPOs aren't in the business of recovering brains, they're in the business of recovering organs, but we know some organizations, whole body programs and tissue banks that might do it for us, blah, 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 blah. You might have to get the it could come any time of the night and they say, oh, I can't do that. I leave at 5, and that's a problem. So I think for researchers, if you come to organizations like us and you need these hard-to-get tissues, please be flexible. This is not like shopping at VWR. Improvements needed in holding shipping conditions media. A lot of people talked about that yesterday. Again, stakeholders that we work with, usually the scientists have defined methods that work for them, or we borrow a little bit from the OPO world and use media and conditions that work for solid organs. But more work needs to be done. Transplant surgeons are doing more with less. So what that means is, what we're seeing is even though organ donation numbers are going up, the available organs for research, particularly livers, is going down. When we get a call from an OPO, sometimes it's a case that's presented to us, they're working it up for donation. We know ahead of time it may go for transplant or it may not. They'll call us back after they've made the offer, and they'll retract it. So in other words, they've found a recipient. So those retraction numbers are going up. Recovery methods for procurement, processing, preservation, and short-term storage are not yet harmonized across stakeholders. Again, something that the field needs work a little bit harder to achieve. Last thing, International Organization for Standardization, I mentioned early, they have a biotech working group that is addressing some of these problems. If you take a look at their website, they do have methodologies and standardizations that we're working on for things like cell counting, evaluating performance of nucleic acid quantifications, et cetera, et cetera. Take a look. There's things there that may be helpful to you. Some examples of non-transplant organizations that provide human tissue, of course, NDRI is one of them. There's some other organizations here. I can't speak to the quality of these different organizations. I'm aware of them. They're competitors in the space. Some of them actually source from us as well. So again, hopefully this information is all helpful to you. Just trying to give a broad landscape of the field, the standards, what they're intended to do, and how we're going to have to work together to address some of these gaps. All right, thank you.